Hello and welcome back to Biology 101. In today's video, we will be exploring the fascinating process of DNA replication, the mechanism by which a cell duplicates its genetic material before cell division. Now, before we dive into the process of DNA replication, it's important to first understand the structure of DNA itself. DNA, or deoxyribonucleic acid, has what we refer to as a double helix structure. This means that it is composed of two long strands that run in opposite directions, or what we call antiparallel. Each strand of DNA is made up of monomers called nucleotides, which consist of three components: a deoxyribose sugar, a phosphate group, and a nitrogenous base. The four possible nitrogenous bases are adenine, thymine, cytosine, and guanine. The sugar-phosphate backbone of DNA forms the outer structure of the double helix, while the nitrogenous bases are located in the interior of the helix, where they pair with complementary bases from the opposite strand. These base pairs follow specific rules. Adenine pairs with thymine, and cytosine pairs with guanine. This complementary base pairing is crucial for accurate replication of DNA, and we'll be going into this later in this video. Now that we have an understanding of DNA structure, let's examine the process of DNA replication, the actual mechanism by which the DNA molecule is copied. DNA replication begins at specific sites on the DNA molecule known as origins of replication. These are the points where specialized proteins bind to the DNA and initiate the unwinding process, creating what is known as the replication bubble. In eukaryotic cells, there are multiple origins of replication, allowing for the efficient copying of long strands of DNA. Each end of the replication bubble contains what we call a replication fork, which is the region where the DNA is actively unwound and being replicated. Now let's take a detailed look at the key proteins and enzymes that facilitate the replication process. In general, these molecules work together in a highly coordinated manner to ensure the accurate duplication of the genetic material. The first enzyme we encounter is helicase, which plays a crucial role in the unwinding of the DNA. Helicase functions by separating the two strands of the DNA helix. Think of it like a zipper unfastening a jacket. As helicase moves along the DNA, it breaks the hydrogen bonds between the base pairs, unwinding the DNA and creating the replication fork. This process exposes the individual strands of DNA, which will serve as templates for the new strands. Once these strands are separated, they are in a highly unstable state. If left alone, the single-stranded DNA could easily reanneal or snap back together, halting the replication process. To prevent this from happening, single-stranded binding proteins, or SSBPs, bind to the exposed single-stranded DNA, stabilizing the strands and ensuring that they remain separated. These proteins essentially act as scaffolding, maintaining the integrity of the separated strands and allowing the replication machinery to function smoothly. As helicase unwinds the DNA, it creates tension ahead of the replication fork, similar to twisting a towel, which becomes increasingly tight as you twist it. This tension can cause the DNA to become overly coiled, potentially leading to damage. To relieve this strain, topoisomerase comes into play. Topoisomerase is an enzyme which works by making temporary cuts in the DNA ahead of the replication fork, thereby relieving the tension. Afterwards, it reseals the breaks, allowing the DNA to continue to unwind without becoming overly strained. Now, at this stage, Primase plays a key role in preparing the DNA for replication. DNA polymerase, the enzyme responsible for building the new DNA strand, cannot begin adding nucleotides on its own. It requires a starting point, and this is where Primase comes in. Primase synthesizes a short RNA primer that provides a free 3' hydroxyl group, which DNA polymerase needs to begin its work. This primer marks the starting point for the DNA polymerase to begin building the new strand. Next, we have DNA polymerase III, the primary enzyme responsible for synthesizing the new DNA strand. Once the RNA primer is placed, DNA polymerase binds to the primer and begins adding nucleotides in the 5' to 3' direction, complementary to the template strand. As it moves along the template strand, DNA polymerase III continues to add nucleotides, elongating the new DNA strand. It does this by matching the appropriate base pairs, A with T and C with G, to form a new strand that is complementary to the template. However, due to the antiparallel nature of the DNA, replication occurs differently along the two strands. You can imagine DNA like a two-laned highway. One lane is moving in the direction of traffic, and the other is moving against it. The two strands of DNA behave similarly. They run in opposite directions, which creates a challenge when it comes to replication. Let's start with the leading strand, which runs in the 3' to 5' direction relative to the replication fork. This strand can be synthesized continually, just like laying down a smooth, uninterrupted road that follows the direction of traffic. As the replication fork opens up, DNA polymerase III, the enzyme responsible for adding nucleotides, moves in the same direction as the unwinding DNA. It continually adds nucleotides to the growing strand without interruption, ensuring that the leading strand is replicated in one long stretch. Now, the lagging strand runs in the opposite direction, from 5' to 3' relative to the replication fork. This is where things get a little trickier. Since DNA polymerase III can only add nucleotides in the 5' to 3' direction, it can't directly replicate the lagging strand, much like trying to build a road against the flow of traffic. Instead, the lagging strand is synthesized in fragments, which we call Okazaki fragments. Think of it like a construction crew building sections of a road backwards. They lay down a piece of road, then stop, go further back, and then lay down another piece. For each Okazaki fragment, Primase places a small RNA primer to mark the starting point. Then DNA polymerase III adds nucleotides to the fragment until it reaches the end of the previous one. After all the fragments are built, DNA polymerase I replaces the RNA primers with DNA nucleotides, ensuring the entire sequence is made of DNA. In summary, the leading strand is replicated continuously, following the direction of the replication fork, while the lagging strand is built in short fragments, each one moving against the fork. But just like how a construction crew ensures that the road is complete, even in the opposite direction, the fragments are eventually joined together to form a continuous and accurate new DNA strand. Now, overall, DNA replication is a fundamental process that ensures the faithful transmission of genetic material during cell division. It is essential for growth, repair, and reproduction, making it one of the most crucial processes in biology. So, what did we learn in this video? Well, firstly, we learned about DNA structure and base pairing, and we know that DNA consists of two antiparallel strands, each made up of nucleotides. The nitrogenous bases on each strand pair specifically. Adenine pairs with thymine, cytosine pairs with guanine. This complementary base pairing is essential for accurate DNA replication. Second, there are several key enzymes involved in DNA replication. Helicase unwinds the DNA, while single-stranded binding proteins stabilize the separated strands. Topoisomerase alleviates the tension caused by unwinding, and Primase adds short RNA primers that allow DNA polymerase III to start building the new DNA strands. Third, we learned about the differences between the leading and the lagging strands. The leading strand is synthesized continuously in the same direction as the replication fork, while the lagging strand is synthesized in short fragments known as Okazaki fragments in the opposite direction. These fragments are later joined together by an enzyme known as DNA ligase to form a continuous strand.